This is going to be the second lecture of Unit 3 on sensation and perception. We will cover all of the senses in this unit, and today we are going to cover the eye. Before we talk about how the eye works, let's talk about the process of transduction. With sensation, there's this transformation of the stimulus energy into the neural impulse, which we talked about to be sensation yesterday. Transduction is when you take that stimulus energy and you process it into something that is meaningful to you. So for example, the light that you see is turned into a neural message, and then the brain processes it as what you consciously see. And we are converting one form of energy to another on a second by second basis. This is not just for sight. It can be for sound, smell, taste. Transduction is just when the chemical energy is transformed into something that is meaningful to us. With the physical characteristics, you have wavelength and intensity that help us to determine our experience of light. The wavelength is the distance from one wave peak to another. And the wavelength is important for determining the hue and the color, like blue or green. Whereas intensity, or the brightness of light, is determined by the height of the wave. So a long wavelength produces reddish colors, whereas a short wavelength produces bluish colors. Now, how do we determine, or how do we perceive wavelength and hue? How does the eye work? I would recommend sketching this out in your notes. So we will go through each of these terms, but I'm going to run through really quickly how the eye works before we get into the definitions of each thing. So let's say you're looking at a candle. How do we perceive, how does this candle get from our vision to our brain? The first thing that happens is light enters the cornea. The cornea is that transparent coating that protects our eye and helps bend light to focus. Once light has passed through the cornea, it then goes to the pupil. The pupil is a hole in our eye. It is an adjustable opening. And the size of the pupil is regulated by the iris. The iris is a muscle that either dilates the pupil or constricts the pupil. The iris allows light to come into the pupil, or it shuts light out from the pupil. So if you look in the mirror, the pupil is the black spot in your eye, whereas the iris is that colored muscle that controls the pupil. Behind the pupil is a lens. If you think about a contact lens, the lens helps to focus the image of what we are seeing on our retina, which is in the back of our eye. The retina contains our visual sensory receptors. And if you look at this little bump out right here, that is the fovea. The fovea is located on the retina. The fovea is the central point of focus. The fovea focuses. Fovea focuses. The fovea is where our cones are located. We'll get to cones in just a second. If you're focusing on this PowerPoint slide, you can tell me what color it is, you can tell me the words that are there, but I want you to not directly look at, but look at the things that are in your peripheral vision. You can probably make out their color, but they're going to be a little bit fuzzy. That's because those items in your peripheral vision are not focused on your fovea. Once the light hits the fovea, activates the cones, it goes to the optic nerve. The optic nerve takes the message, takes the chemical energy, the light, from the eye to the brain, specifically to the thalamus, and the thalamus sends it to the visual cortex. Now, where our optic nerve leaves the eye and goes to the brain, we will find our blind spot. So again, going through parts of the eye, the cornea is the transparent tissue where light enters the eye. The iris is the muscle that expands and contracts to change the size of the pupil. The lens helps focus the light rays on the back of the eye, which is the retina, and the retina contains our receptors. Now the lens, which is that transparent coating, which you will see right here, changes the image of our eye. This is the process of accommodation. Accommodation occurs when the shape of the lens changes to focus on things that are far away or things that are close up, like you see in the bottom diagram. If you are nearsighted, your eyeball is actually misshapen. It is not a circle. Your lens is more like an oval. The eyeball focuses light on the front of the retina instead of the back of the retina if you're nearsighted. If you're farsighted, your eyeball, the lens is actually shortened. The light rays reach the retina before they have focused the image. So that's near and farsightedness. Now, as far as the retina goes, like I told you, it contains these receptor cells, which are known as rods and cones. And the rods and cones activate something called bipolar ganglion cells and bipolar cells and ganglion cells. So what happens when the light hits the retina? Light enters the eye and triggers these photochemical reactions in the rods and cones. You can see the red items are cones and the green items are rods. Yes, there are more rods in the retina than cones. Once these photochemical reactions in the rods and cones are focused on the back of the retina, there's a chemical interaction that takes place that activates these bipolar cells. All of the rods and cones, as you can see here, are linked up to these bipolar cells. The bipolar cells are linked up to ganglion cells, which all come together right here to create the optic nerve. Light essentially breaks the outer surface and then buries into these receptor cells, activates the rods and cones. The information or the light leaves the ganglion cells, goes to the optic nerve. And where the optic nerve leaves the eye and goes to the brain, you have this blind spot. Essentially, there are no receptor cells located in the blind spot. And again, the fovea is the central point of focus. In the fovea, you've got mostly cones that are located there. So what's the difference between those rods and cones? Cones help determine color vision, whereas rods help determine black and white vision. There are more rods than cones in the retina, in the eye. And cones tend to cluster around the fovea. So cones cluster around the fovea, whereas rods are found all throughout the retina. Now, how do we process this information visually? How does our brain process it? Again, the optic nerve connects to the thalamus. The thalamus is that sensory switchboard in the middle of the brain. And the thalamus is going to send that information to the visual cortex. The visual cortex, is in the back of the brain, which would be in what lobe? The occipital lobe. And remember, information that is perceived with your left eye is processed on the right side of your brain, and information received with your right eye is processed on the left side of the brain, like we talked about with split brain patients. Now, as far as perception of vision, there's some different theories that you're going to need to know, starting with the feature detection theory. This theory says that there are nerve cells in the visual cortex, in the occipital lobe, that respond to specific features of shapes or of things, basically edges, angles, and movement. So the visual cortex has these neurons, these feature detector neurons, that allow us to say, that looks like a sharp edge, or perhaps perceive a threat, then, oh yes, something is coming towards me, or something is moving away from me. That's the feature detection theory. Whereas the parallel processing theory of vision says that we process several different aspects of the stimulus simultaneously. This is called parallel processing. Basically, what this theory says is that the brain divides a scene into different divisions: color, depth, form, movement. And once the brain divides that visual scene, it's giving us information as to, that is a bird. The bird is brown and black and gray. That bird is moving. That bird has a specific type of form and depth. And this is all happening at once. So for example, with the parallel processing theory, if you see a familiar face, what's happening is the brain is integrating information that the retina sends to the visual cortex. It's comparing it to stored information in your memory, in your hippocampus, and it enables you to recognize someone as someone that you know. Now, as far as color vision goes, there's some different theories of color vision. The first is the trichromatic theory. This was created by Young and von Helmholtz, and they suggested that the eye must contain at least 3 different receptor cells that are sensitive to 3 colors: red, blue, and green. Trichromatic theory: red, blue, green. When we stimulate a combination of these cones, remember cones are color, we see other colors. So for example, there's no receptor cells for yellow, but when red and green are stimulated, we see yellow. If you are colorblind, it is a genetic disorder, and basically what the color vision or the trichromatic theory would say for colorblind people is that they lack in functioning red and green cones. Their red and green cones are not functional, and so therefore they are colorblind. The last theory I want to talk about is the opponent process theory, which says that we see opposites of colors. So for example, the opposite of red is green, the opposite of blue is yellow, the opposite of black is white. And after leaving our cells or our cones, visual information is processed in terms of their opponent color. So if you were to gaze at the middle of this flag for about 30 seconds, when it disappears, stare at a dot or something white, and you will see the British flag. That's because the green and yellow flag, the opponent colors of green and yellow, would be red and blue. Therefore, you would see the British flag. And what I'm going to do now is play a video for you that you might want to link to yourself. It might work better, but we'll see if it'll work here.
After image in your brain, what you need to do at this point is to stare at the dot in the center of the photo. And keep staring at the dot. Don't move your eyes. Keep staring at the dot. The photo is strangely colored. That's all to do with the demo. So just keep staring at the dot. And in about 20 seconds time or so, the photo is going to be replaced by a black and white version. But you're going to probably see some color in that photo. So keep staring at the dot. That's good. Don't move. Keep staring. There we go. If you move your eyes quickly off to the side and then back to the dot, you should see the black and white photo, and then the colour should reappear when you get back to the black dot. Probably starting to fade now. You're all probably familiar with the idea that a TV screen makes colours using red, green and blue pixels at each point. Each colour we see is a particular mixture of these colours. Our eyes aren't TV screens, of course, but they do contain 3 types of colour-sensitive receptors known as cones. One sensitive to red wavelengths of light, one green, and one blue. So the way the brain sees different colours is by looking at the relative activity across 3 types of cones. So, when you stared at the strangely coloured building, you are in fact tiring out or fatiguing the 3 types of cones in different ways across the photo. The area of grass was painted mauve, for instance, and this fatigued the red and blue cones that have a view of the grass more than the green cones. When we now replace the strange photo with a black and white photo, we are in fact seeing colour created by different levels of fatigue in each of the cones. The brain only knows about different colours by looking at relative activity across the 3 types of cones. Remember I said if you moved your eyes when looking at the black and white photo, the colour after image disappears? Well, this is interesting because this tells us that the structure of the scene you use to see the after image must be similar to the structure of the scene you use to tire the cones out with. Any old building won't do, and the same building placed in different positions on the retina won't do either. This may explain why we don't see colour after images all the time, because our eyes are continually moving around.
Okay, that's the end of the second lecture. Again, make sure that you know exactly how the eye functions and each of these parts of the eye.